Hello friends, I am Rajesh Pal and welcome to my channel Nothing But Biology. Today we will discuss about transport through the plasma membrane. Transport through the plasma membrane. Actually, the transport of plasma membrane can be divided into two parts. One is passive transport and another is active transport. One is passive transport and another is active transport. Passive transport means where energy is not required. Where energy is not required. And active transports mean where energy is required. Today actually we discuss the passive transport. And in the next video we will discuss about active transport. So this passive transport can be divided into two parts. One is simple diffusion and another is facilitated diffusion. And facilitated diffusion can be divided into two parts. One is carrier protein mediated and another is channel protein mediated. This channel protein may be ion channel or ligand-gated channel. This is the main types of passive transport, and we will discuss about passive transport. What is passive transport? Passive transport is the type of transport where energy is not required. It means ATP is not required. This passive transport have many characteristic features. One is the transport of things from high concentration to low concentration. Another is not required ATP. It means energy is not required. Not follow the concentration gradient. Not follow the concentration gradient. Not follow the concentration. Sorry. Concentration gradient আমরা আলো করেছি, follow the concentration gradient but not follow the saturation kinetics. Not follow the saturation kinetics. May require or may not be required receptor. Receptor may or may not be required. So what are the main point? Follow the concentration gradient mean high concentration to the low concentration. Movement of things from the high concentration to the low concentration. Not require ATP. It means energy is not required. Let's come to the point. Not follow the saturation kinetics. What we actually understand from this point, let's discuss about this point. If we think this is the inside of a cell and outside of a cell, there are 60 atoms of glucose and there are 20 atoms of glucose. What will happen? So high concentration to low concentration movement is known as passive transport. 60 to 20 movement will occur. But when saturation kinetics mean when the number of glucose is similar in inside and outside of the cell, is similar in inside of and outside of the cell, then no movement will occur. Actually, this point wants to mean that it does not reach the equilibrium state. The difference of molecule, difference of ion always happen, and the point receptor may or may not be present. And let's come to the point simple diffusion. Passive transport is divided into two parts. One is simple diffusion, another is facilitated diffusion. Let's come to some point simple diffusion. Simple diffusion. What actually happened in simple diffusion? During this process, the molecule can transport through the plasma membrane, can transport through the lipid bilayer of the plasma membrane easily. In this case, no protein is needed. In this case, no protein is needed. If any kind of thing, what is this? What is this? Hydrophilic, and this is hydrophobic. If the thing is water soluble or water like material, then it can easily pass through the lipid bilayer. This process will called simple diffusion, and the point will come the facilitated diffusion. Facilitated diffusion. FASCILITATED, facilitated diffusion. What actually happened in facilitated diffusion? In this type of diffusion, ion or nutrient can move. Ion, nutrient, any kind of ligand can move through the plasma membrane by the help of protein. This facilitated diffusion can be divided into carrier protein mediated or channel protein mediated. Carrier protein mediated, and channel protein mediated. Carrier protein ক্ষেত্রে what happened? In case of carrier protein, what happened? Draw. Wait. We think this is I am drawing some cluster. actually what we are discussing about, is carrier protein. Carrier protein. What happens in case of carrier protein? When this is the structure of carrier protein. When any kind of thing, things or nutrient, can be any kind of ligand, binds with this point, what will happen? Conformational change will occur. And this enter to the inside of the cell. It is outside of the cell and this is inside of the cell. When any kind of ligand, hormone, nutrient, ion can bind, then conformation change will occur in case of carrier protein, and if the ligand, ion, nutrient can enter to the inside of the cell. Actually in case of glucose happen, group transporter. When group transporter needs glucose transporter, I think this is a group protein, and what will happen when glucose bind with the this is a glucose, I think this is a glucose, and this is group protein, and this is glucose. And what will happen? Conformational change will occur and glucose will enter to the cell. Then the structure of, when conformational change will occur, then the structure of protein will like this. And the glucose will enter to the cell. This is carrier protein mediated transport. And let's come to the point, channel protein mediated. When two protein position with together, then a pore or channel found. Through this channel, element can move. This channel, element, hormone, any kind of ion can move. This channel protein can two types. One is voltage-gated ion channel and another is ligand-gated ion channel. In case of voltage-gated ion channel, what happens? Every cell have different voltage gradient. This is found in Na plus and K plus channel protein. ठीक है? I think there is Na plus is high, and now actually Na plus is high in the outside of the cell and K plus is high in inner side of the cell. When actually we draw a graph. The resting membrane potential. What is? The cell have a voltage different, which is known as resting membrane potential. This, I think many type of cells have many type of voltage gradient. I think this is a cell where voltage gradient is -20, and when it will increase the action potential, when it will increase the action potential at a point Na plus, at a point Na plus will enter. When Na plus will enter? When the cell is resting membrane potential, then all the I think this is Na plus channel and this is K plus channel. Na plus channel and this is K plus channel. When the resting membrane potential reach to the or action potential reach to the point plus 20, then Na plus will enter. What happened? This thing will remove and Na plus will increase rapidly. Na plus will enter rapidly inside the cell. What will happen? This Na plus plus. This Na plus will increase the positive charge of the cell. And at this point, difference of voltage gradient will increase, and at a point K plus, when I think it is 40 plus 40, when voltage gradient is plus 40, then the K plus will remove from the inside of the cell. From the inside of the cell, what will happen? Actually, K plus, the closing point, door is inside the cell, and door will open, and the K plus will remove from the cell rapidly at this point at plus 40, and then rapidly this is called depolarization. When increase of action potential is known as depolarization, and when K plus rapidly moves from the inside to the outside of the cell, and the action potential will decrease and comes to the point minus 20, and this is repolarization. Depolarize. Understand? If you don't understand, please comment, and I will again make a video for you. And this is the Na plus and K plus channel, which actually passive transport by the help of channel protein. And let's come to the point. Ligand-gated ion channel. What actually happened in ligand-gated ion channel? When a ligand binds in this point, then the ligand comes to the point and at least comes to the inner side of the point. Ligand-gated. Then comes to the point. This is the ligand protein. This is the channel protein, which is actually ligand-gated, and in previous we actually talk about the channel protein, which actually acts like voltage gated ion channel. In ligand gated, the ligand will bind and slowly it will enter to the cell, or in any case it can go outside of the cell, it can go outside of the cell, it can go outside of the cell from inner side of the cell. In same way This is actually what? This is actually passive transport of plasma membrane. What we discussed today? Today we discuss about the passive transport of plasma membrane.